Let's move on to our second objective here, which is to discern between our body's different mechanisms of energy storage and energy usage. So what are the other ways that we can regulate the way that our body uses and stores energy? So this brings us to talking about our other biomolecules. So far, we have been describing glucose oxidation as the main source of cellular respiration. What does that mean? So far, we've been saying that when we break down glucose and liberate ATP, this is the main way that our body can make energy. But there are other ways, other substrates that we can break down to make ATP as well. Glucose is going to be the most reliable, the most abundant, the most efficient, but we can also use lipids or fats, right? So glucose is going to be our most abundant, but we can also use lipids to make a lot of ATP, and then proteins, we're going to make ATP as well, but a very minimal amount. And proteins aren't going to be broken down unless the body is in an absolute crisis, so to speak, or it is limited in terms of the other more reliable sources. So if we're completely out of carbohydrates and completely out of fats, right, subcutaneous fat, body fat, fat coming in from the diet, then we're going to start tapping into using protein, which is really what is making up our muscles, right? So I really want to stress this, that while we can use lipids and fats alternatively, proteins are going to be a last resort. We really don't want to start burning muscle unless we absolutely have to. All right, so this is how all three of these molecules can be broken down, and this is how the processes are really tied together, such that if we run out of carbs, we can tap into fat. If we run out of carbs and fats, we can tap into proteins, right? So all of the processes are connected in terms of breaking it down to yield ATP, okay? So we've talked about the process in the middle already. We talked about a molecule of glucose being ingested through our diet, that undergoing the first stage, which is glycolysis, yielding pyruvate. From pyruvate, we go to the linking step. So this would be considered the linking step here. And then from the linking step, we make acetyl-CoA, and that acetyl-CoA enters the Krebs cycle, which would be considered the second major step. From there, the Krebs cycle is continuously running in a cyclical fashion and is generating all of the substrates that we need to drive oxidative phosphorylation, which is the third and most reliable, most efficient way of breaking down glucose, or a stage of breaking down glucose to make ATP, okay? So this is what we have said so far, and this is only considering glucose or carbohydrates as the source or the substrate that is being broken down. Alternatively, we can break down our fats. So we know that fats, specifically triglycerides, are made of two components. There's a glycerol head and the fatty acid tails. Both of these components can be broken down. They can enter glycolysis, okay, and they can drive the breakdown of fats in that way. They can also enter the Krebs cycle in the form of acetyl-CoA. Additionally, the fatty acids can enter directly into oxidative phosphorylation as well. So some of the breakdown products from triglycerides can enter the various stages that we've discussed and help to be substrates to utilize in order to make ATP. On the other end, and as we said, this is a very rare or last resort scenario, we can tap into proteins, break down proteins into their smaller components, cleave those peptide bonds, and liberate the amino acids, which are the individual units in proteins. And then that can be broken down as well. That can be converted into pyruvate and enter the linking step here. That can be converted into acetyl-CoA and enter the Krebs cycle, or that can be converted into some other substrates that can directly enter the Krebs cycle as well. All right, so I really want to stress here that we can use other substrates, other biomolecules to break down. They can be fed into the stages of glucose oxidation and help to make ATP with the energy that's being stored in those specific substrates. All right, so they basically can be considered as fuel for the fire, right? Think about them as being wood that we throw onto a fire. The fire is making the energy, but we can put different sources of fuel into that fire to really help us generate energy with different sources in mind. Okay. Let's think about glycogen now. So glycogen is the storage form of glucose whenever we ingest glucose. Let's say we had a really large meal and our body is really well fed. We're not going to break down all of that glucose. We're going to break down just what we need, and then the rest of it is going to get stored up in the liver as well as the kidneys in the form of glycogen, okay? And so this is why when we have a period of fasting, so let's say we are not having a meal for a very long time, we can then go back, tap into the liver, tap into the kidneys, and tap into these additional sources of glycogen, and then reverse the process and make glucose that can be used by our cells to make ATP on the spot. All right, so glucose can leave the cell, but not glucose 6-phosphate. So I'll talk about what glucose 6-phosphate is, but it's really important to understand that only glucose can leave the cell and the other substrates cannot. So we can only directly process glucose in order to use it as energy. We cannot directly grab glycogen. We have to convert it back to glucose, and then it can leave the cell and be used again. Not only the liver and kidney have the enzyme that is needed to convert glucose 6-phosphate back into glucose, okay? So what we really want to say here is that we need to have glucose in its original form in order for it to be used in glycolysis, and we need to have a specific enzyme which is only found in our liver cells and kidney cells to convert the glucose 6-phosphate back into glucose so that it can then leave the cell and supply our cells with energy in a state where we're not necessarily eating or we're not necessarily bringing in more glucose. We can tap into our stores, but we have to convert it back to glucose first. All right, we can also synthesize new glucose using a few other substrates. So glycerol, we mentioned this on that previous slide. If we took a fatty acid, so here's a fatty acid, or here's a triglyceride. If we strip away this glycerol head from the fatty acid tails, we can send this glycerol as a substrate for making new glucose as well. We can also use lactate. So we talked about how lactate was made. Pyruvate was converted to lactate under the circumstances that there's no oxygen being supplied. We can then shuttle that pyruvate off to make lactate, and that can be used to make some new glucose as well. And then under, again, extreme circumstances, we can make amino acids converted back to glucose as well. And these are going to happen mainly in the liver. Really important here, the liver is the only organ, and kidney are the only organs that have the enzymes that are capable to converting some of these additional substrates back to glucose so it can leave the cell and go around to all of our other tissues. All right. We also want to point out that this is not simply the reverse of glycolysis because entirely different enzymes are included. So we go from glucose 6-phosphate, glucose 6-phosphatase, this is the enzyme that is being used, and then we yield glucose. As far as the reverse of glycolysis, it's different enzymes. So here we're using glucose, right? Here's the glucose, and then here is hexokinase, which is a different enzyme, and then we go to that glucose 6-phosphate. All right, so this is happening in the forward, and then this is happening in the reverse. So we really want to stress here that it's not just the reaction happening back and forth. It's entirely different enzymes that are being used to bring about either glucose 6-phosphate or glucose on the opposite end. All right, so here's another illustration of that. Here is this glycogen. This, again, is the storage. Excuse storage form of glucose, right? So this is in liver and kidney. And so what we need to do is we need to convert this glycogen back to glucose in order for it to leave the cell. So we can do that by breaking down the glycogen to glucose 6-phosphate, which is this guy here. And then we're going to need the enzyme glucose 6-phosphatase to convert it back to glucose so it can now leave the cell. And when we say leave the cell, we mean leave the liver cell or leave the kidney cell, and then circulate back to all of our other tissues to be able to supply them with a substrate to make ATP. Now, if we think about the opposite of this process, it's going to be hexokinase. So it's an entirely different enzyme that converts glucose to glucose 6-phosphate, which is allowing it to be stored. So this is the enzyme that's going to help it go into storage, right? Glucose 6-phosphate, and then we can move it up to glycogen. And then the enzyme that's going to be needed to liberate it from storage is the glucose 6-phosphatase. All right. Okay. Now, if we look at the energy storage, most of our energy is stored in the form of triglycerides. I'm sure we already know this from our day-to-day knowledge and experience. If you have too much energy that's being taken in, you're going to start putting on weight, right? You're going to start storing that into your adipose tissue, storing that into your fat, whether it be visceral fat that's covering your organs, or subcutaneous fat that's right underneath the skin. The main storage of our body is going to be triglyceride, which is fat. All right, so there's going to be 3 fatty acids and 1 glycerol. We drew out the structure of this before. So the fatty acid tails and the glycerol head are what comprise a triglyceride. And lipids have more energy per weight than carbs and proteins. That's also super important. A lipid is going to yield us 9 kilocalories per molecule. Proteins and carbs are going to yield us 4 kilocalories per molecule of both proteins and carbs. So what this means is that fats are the most efficient storage of energy. When we break down 1 molecule of fat, 1 triglyceride, we're going to yield more energy, 9 kilocalories on average per molecule that's broken down, versus carbohydrates and proteins, although carbohydrates are more abundant, right? So they're more abundant in our diet as well as in our body, but it's not going to yield us as much energy as if we were to store that up in the form of fat or lipids, okay? And then as we said earlier, the way that this is stored in our body is in adipocytes, which are basically our fat cells. Okay, so you've got fat cells that can be really large. So here's what a fat cell looks like. Here's the nucleus of that fat cell. Here's all of the storage triglyceride in the cell itself. Or it can be a fat cell that's really small if it doesn't have a lot of that triglyceride being stored. And this is why fat takes up more space. As we put more triglycerides into the cell gets bigger and bigger and bigger, and that is what essentially comprises fat. All right, if we think about metabolizing fat, this is called fat metabolism. It's going to include a process called beta oxidation. So if we think about the structure of triglycerides that we just described here, so here are our triglycerides. We can separate them out into their 2 main components, fatty acids, which are the tails, and the head, which is the glycerol head. The glycerol head can undergo glycolysis, so it can be thrown in, again, like fuel into the fire. It can be fed into glycolysis. The fatty acids can do a couple of different things. They can be converted into the coenzymes that get directly shuttled into oxidative phosphorylation, or they can be converted into acetyl-CoA that can be shuttled into the Krebs cycle. So if we split out this triglyceride into its different parts, it can be used as fuel into the various stages of oxidation. Okay. Additionally, beta oxidation. So beta oxidation is the specific process where we convert fatty acids into acetyl-CoA. So here's where we have that beta oxidation taking place. So we essentially make acetyl-CoA, NADH, FADH, that acetyl-CoA can get shuttled into the Krebs cycle, which is happening here. And then the electrons will be shuttled off into the electron transport chain as usual. And then we're going to yield some ketones. So ketones is a byproduct of breaking down fatty acids and shuttling them off via acetyl-CoA into the Krebs cycle. And then these get broken down, these get released rather as a byproduct of beta oxidation. Okay. So this is how we can use fat again to fuel our other oxidative processes. Let's look at proteins and how this can happen with proteins. So again, we have proteins. They can be broken down into their smaller components, which are amino acid. And then from there, we're going to undergo a process that's called deamination. So deamination is basically to remove the amino group from the amino acid. And then what we have left behind is ammonia. So ammonia is going to be released as urea. That's going to be filtered through the kidneys. Lost is going to be the main component of urine, if you think about urine. And what is lost as that waste product? That would be the deaminated form of amino acids, which is that ammonium converted into urea. We're also going to see that that's going to be then shuttled off into keto acids. And then keto acids can directly enter glucose oxidation as well. So they can enter as pyruvate, or they can become acetyl-CoA, or they can be directly shuttled into the Krebs cycle. All right. Again, really important to stress that this is not an everyday occurrence. This is a very rare phenomenon. Very rarely, do we begin breaking down our proteins or our muscle. That's just not the way the body is designed. Proteins are going to be broken down as a very last resort.